Welcome to a brief introduction into CRISPR gene editing. What we're going to do is we're just going to talk a little bit about how CRISPR edits the genes. Now, CRISPR, it's a little bit different from how a prokaryotic gene would generally um, regulate its expression, and a eukaryotic gene for that matter, because most of the times in prokaryotic and eukaryotic genes, the regulation is done at the transcriptional level. And so generally they would do it at transcription. They would change up the transcription factors. They would stop RNA polymerase from binding and DNA polymerase from binding when you were making that mRNA. But with CRISPR, what you're doing is you're knocking out the gene right from the get-go. So it's pre-transcriptional. Even before transcription or anything takes place, we are going to knock out and we are going to change the genome and destroy a gene. And so what happens is we have this thing called our CRISPR. And so this is our CRISPR. And so what it does is what it's made up of a couple different components, but the one that we are interested in is this section right here, and it is made up of protospacers, which is in the purple, and short palindromic repeats. And so what the protospacers are is the protospacers are the viral DNA that we have taken from lysogenic viruses that have infected the cells, and our short palindromic repeats are just our tags so we know what we're looking for. So we have the viral DNA and we have our tags, and we have multiple different viral DNAs and multiple different tags. These are from different viruses that have come in the past. And so what we are going to do is we are going to take the double-stranded DNA. We are going to get RNA polymerase to come on by, and we are going to transcribe this into what we call pre-CR RNA. So we're going to transcribe it into some pre-CR RNA, and this, instead of being double-stranded DNA, now we just have some RNA. And what's going to happen is we're going to get a couple of different things now. And so we are going to get some tracer RNA. They bind and they help search the cell for the lysogenic viruses that we want to kill and the genes that we want to delete. We are going to get Cas9 complex to bind to that as well. And we are going to get RNase 3 to come and bind. And so what we have in this pre-CR RNA is we have multiple protospacers and multiple short palindromic repeats. And so what we're going to do is, what RNase 3 is going to do is going to separate these. So it's basically going to split them apart. And so now we each have a section of pre-CR RNA that is only searching for one viral genome. So we have the pre-CR RNA, we're going to have that tracer RNA, we're going to have the Cas9 complex, and we're going to have this for each section. And so we're going to add a tracer RNA and a Cas9 complex. And so after this is done, we are going to go and scan the DNA. We're going to go scan the DNA using this complex. We'll only show one for simplicity's sake. So we're going to go scan the DNA, scan the DNA, scan the DNA, scan the DNA. And then we are going to go and we're going to find what we're calling a target sequence. And this target sequence is the viral information, DNA or RNA, that is present inside our genome, because once again lysogenic viruses insert their own genes into the cells. They insert their own genes into the genome, and so we're trying to find that genome. And so we find the target sequence, and what we're actually looking for is something called the protospacer adjacent motif, or PAM. And PAM is going to be the thing that we actually look for. It's going to be adjacent to it, adjacent to the target sequence. And so we're going to go scan, we're going to go scan, and then the pre-CR RNA is going to go recognize the PAM. It's going to go bind to it. And then what this complex is going to do, basically, it's going to find the viral DNA where it is. It's going to go find it, be like, hey, it's right here where that pink is. And what it's going to do is basically going to snip off and cause a double-stranded break in the DNA. And double-stranded breaks in DNA are bad. A lot of the time, they completely destroy a gene. But in this case, because we know that this is a viral gene and we do not want this gene, we want to destroy it. And so we make this double-stranded break, and then DNA polymerase is going to go come along and just be like, oh no, there's a double-stranded break. Let me go get some information and just kind of guess and to just kind of repair it. And so a lot of the times that will knock out that viral gene. And so, yeah, that's basically how CRISPR works. Is it goes and it knocks out, it creates a double-stranded break where the PAM is. This isn't really going to affect the promoter or the coding region in most cases. So it will still transcribe, but it's broken at this point. So whenever it makes that mRNA, it's going to not work. It's not going to function as it should. And so yeah, when we make that break and then we repair it, that gene will lose function in the DNA. And then that's basically how CRISPR is going to work and going to destroy viral DNA.